this final video on the chapter of DNA, we'll be looking at RNA, its structure and functions, and then finally we'll move on to protein synthesis. So with regards to RNA, you need to know the location where we find RNA. There's 3 types of RNA, but you only need to know about these 2. So the first one is mRNA, that stands for messenger RNA, and that is found in the nucleus, but it will also leave the nucleus and go out into the cytoplasm. And then the second one is tRNA, and that stands for transfer RNA, and tRNA is found in the cytoplasm of the cell floating around. And then the last one is ribosomal RNA, or rRNA, and that is found in the ribosomes. And the function of rRNA is actually just of reading the amino acids and then linking them together, as you'll see a bit later on when we look at protein synthesis. So once again, we will look at protein synthesis and look at RNA and its function in protein synthesis just a bit later on in the video. Then the structure of RNA, very similar to that of DNA. The only 2 differences here is that RNA is a single-stranded molecule, whereas DNA is double-stranded, and then it also comes in with the nitrogenous base that differs with RNA. But the nucleotides are still the same: sugar, a phosphate, and then the nitrogenous bases. So the nitrogenous base that is different here is uracil. With DNA, uracil would have been thymine. Now uracil takes over the place or the role of thymine in RNA. And then a stick diagram of mRNA and tRNA to illustrate their structure. So let's quickly look, a quick recap at DNA again. So DNA is a double-stranded molecule, a double helix structure, as you can see on the left, and then the nitrogenous bases there are adenine, guanine, cytosine, and thymine. And adenine always bonds with thymine, always together, and then guanine with cytosine for a good couple. Now with RNA, it is a single-stranded molecule, as you can see, so this is just a helix structure, whereas DNA is a double helix structure. And then the nitrogenous bases, adenine is still there, guanine and cytosine, but thymine is replaced with uracil. So how do they bond with each other? You might ask. Adenine will bond with uracil, that uracil purely replaces thymine, and then guanine will still go with cytosine. So if we look at DNA, a DNA strand, let's say we've got a strand over here, and we've got adenine, guanine, cytosine, thymine. Let's make it a guanine. So what it will bond with now, so this is a DNA strand, remember? So the complementary strand to this one, there'll be a thymine, a cytosine, guanine bonding with that cytosine, thymine will bond with adenine, and guanine with cytosine. And then holding these 2 strands together, in between there will be weak hydrogen bonds that will break or unzip rather as DNA is replicated, or in the case of protein synthesis, as we'll see a bit later. Now with RNA, RNA is going to come in and copy DNA in protein synthesis. So let's have a DNA strand. So pretend that this one was now unzipped. So I'm just going to use this one's example. So we had a thymine, a cytosine, a guanine, a adenine, and that was a cytosine. Let's add one more, let's say thymine. So let's see what the complementary RNA strand would then be. So if RNA were to come and read this strand, thymine would bond with adenine, as is the rule. Cytosine with guanine, guanine with cytosine. Adenine will now bond with uracil and not thymine. Why? Because this is a Na strand, and then cytosine with guanine, and then thymine will bond with adenine. So let's say there's a thymine, you're not going to start having weird things happening because adenine will bond with uracil and RNA. If there's a thymine, RNA is still able to read that that should be an adenine that is going to be copied. But if there's an adenine on the DNA, it will still bond with uracil. If that does not make sense yet, don't worry, we'll look at this in more detail with protein synthesis. This is just a quick introduction to the whole RNA, DNA strand being copied. Let's look at the general structure of RNA, the two RNAs, tRNA and mRNA. So mRNA is very easy to draw. It just is a single strand, as you can see here. So mRNA, if you had to draw it, just draw a single strand, and then you can just draw the nitrogenous bases in, for example, let's do, that is not correct because this is RNA, so that should actually be a uracil. See what happens if you work very quickly. Okay, so for example, that would be an mRNA strand. Then tRNA has always reminded me of something that looks like a chicken's foot. I don't know why, I just always think of a chicken's foot when I see it. So tRNA has got like this hairpin bend structure with these protruding sides that come out. So that is how you would draw a tRNA. And then very important, at the end of tRNA, there is something that we call an anticodon. So an anticodon obviously is the anti of something. So the codon that it is the anti to, if that makes sense, is on the mRNA strand. And that comes in when we read the nitrogenous bases on these strands. We read them in threes. And these groups of 3 nitrogenous bases are known as a codon on the mRNA. So on the tRNA, when we read the nitrogenous bases on it, it is known as an anticodon, and they also occur in groups of 3. So basically what happens is that the mRNA, the messenger RNA, brings the message from the nucleus with these codons, and then the anticodon of tRNA comes and reads it. So on the one point of the tRNA, you will find these 3 nitrogenous bases, and then the anticodon will try and find the match of the codon. So in this case, this codon is GAG, guanine, adenine, guanine. So then the anticodon that would fit on this one would be cytosine, uracil, cytosine. So that's how we read these nitrogenous bases, just so you know. So on the one end, you've got the anticodon, but then attached to the transfer RNA is an amino acid. So there are 20 amino acids known. And there's a table with them over here, alanine, arginine, and all of these listed up until valine. So attached to the tRNA will be one of these amino acids. Now these amino acids will eventually, if you remember your grade 10 work, all of these amino acids, if there's a chain of more than 50, they will form a protein in whichever combination that the mRNA strand brings. Now with lots of research being done, we know which codons will bring what amino acid to the game, and there is a table like this in the answer series notes, and then they can also give you this kind of table in an exam paper. But basically it has a predetermined list of what codons will link to what amino acid. For example, the codon uracil, uracil, uracil will be for the amino acid PHE, and PHE is phenylalanine. And let's look at another one. Let's say ACU is for THR. THR is there and it's called threonine. So you can see how these 20 amino acids will have various combinations that can be used to produce the amino acid. If you don't understand that yet, don't worry too much. We'll get into more detail now as we go into protein synthesis. So protein synthesis is the making of proteins, and both the nucleic acids DNA and RNA are involved in this process. Protein synthesis occurs in two stages. So the first one is transcription and the second one is translation. Now it's very easy to get confused with the two words, so the way that I remember it is that C, transcription, the C comes before the L, translation. So C comes before the L, so thus transcription is the first step of these two stages. Now, in your exam guidelines, they've given you an exact outline of how this process occurs, as well as with translation, and this is what you should study when you are studying protein synthesis, because this is exactly out of memos from previous exam papers. So it'll tell you point for point what you need to write down and understand out of these two processes. So transcription is the first one, and this takes place within the nucleus of the cell. And basically what happens here is that DNA will unwind, very similar to DNA replication. The DNA strand unwinds, and then the weak hydrogen bonds break so that the DNA can unzip, so it exposes those nitrogenous bases. And then you'll have two separate strands of DNA that are formed. The one strand, very important here, is only one strand is used as a template, whereas with a DNA replication, both strands act as a template. With protein synthesis, only one of the DNA strand acts as a template, and then that template is then used to form the complementary mRNA strand. And how that is done is by the RNA in the nucleoplasm have free-floating RNA nucleotides. So remember, the nucleotide is the nitrogenous base that is connected to the pentose sugar that is connected to the phosphate. So there's free-floating nucleotides in the nucleoplasm with that specific nitrogenous base that will be complementing the DNA template. So the mRNA is complementary to the DNA, and now the mRNA has a coded message for protein synthesis. The mRNA moves from the nucleus to the cytoplasm and attaches to the ribosome. So let's quickly look at this first process. So in the nucleus, you've got the DNA strand, and basically the DNA strand then unwinds, it unzips, exposing the nitrogenous bases. Then only one side of the DNA strand is used as a template. So now mRNA has free-floating, the RNA is free-floating nucleotides, and these nucleotides are going to try and find their buddy. So in this case, on the DNA strand, there's a cytosine, thymine, cytosine, cytosine, guanine, cytosine, adenine, and so it carries on. So now the free-floating RNA nucleotides are going to see where they can match up. So if there's a nucleotide that contains the nitrogenous base guanine, it will bond with the cytosine. If there's one with an adenine, that can bind with the thymine. Cytosine with guanine, cytosine with guanine. So you can see how this mRNA strand is now starting to get built up. And remember, with RNA, the thymine is replaced with a uracil. So if there's an adenine on the DNA strand, that will then bond with a uracil and not a thymine. So as soon as this strand is built up, it will then leave the nucleus out of the nucleopore and go into the cytoplasm and head straight for a ribosome. It then attaches to the ribosome, and that is the end of transcription. So please remember, the reason why we are using mRNA is because DNA cannot leave the nucleus. So we need that messenger RNA that will then be able to take that message, the coded message of the DNA, so that a protein can be made. So that is the end of transcription, and that then brings us to the next process, which is translation. So what happens in translation is that each tRNA carries a specific amino acid. We know that because if we look at the tRNA structure, remember at the bottom you'll have the anticodon, and attached to it will be the specific amino acid. When the anticodon on the tRNA matches the codon on the mRNA, the tRNA then brings the required amino acid to the ribosome. You don't need to know the names of these amino acids, because if you remember, they are quite big names, so it's not expected of you to memorize those, but you do need to know the process of transcription and translation, and that the tRNA will bring a specific amino acid. Then the amino acids become attached by peptide bonds to form the required protein. So let's look at this process. Now you've got that mRNA string that is inside the ribosome. So let's draw that specific mRNA strand that is there. So we've got cytosine, guanine, 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 uracil, and then once again guanine, guanine, uracil. And so it carries on. Remember, we read these nitrogenous bases in groups of 3, known as a codon. This tablet is horrible with writing today. So then the next guanine, guanine, uracil will also be a codon, and then you will have those free-floating tRNAs carrying an amino acid that is going to come in and see where they match up. So now the anticodon, they will have various tRNAs. So this one might be for adenine, cytosine, guanine, cytosine. So it doesn't match anywhere here, so it just goes back. But then you might get a tRNA that has the correct anticodon. So in this case, it would have to be, for this first section, cytosine, guanine, guanine. So it would have to be a guanine and then cytosine, cytosine. And attached to that one, it will have a specific amino acid. Let's quickly look. So in this case, the specific amino acid, you look at the mRNA codon, so CGG. So let's look at that table and see what CGG is. CGG is over here. So just for example, reading this, the first letter is a C, so you look on this side. So this is the row you would look in. Then the second letter is a G, so you would look in this column, and then you would look for the third letter, which is another G, so you would look for it here. So this is the block that you would look for CGG in, and there it is, CGG, and it is the amino acid arginine. So then you would have a tRNA carrying that specific anticodon, and that specific anticodon will bring in arginine. And what will then happen is that tRNA will then drop that amino acid there, and so it carries on as the strand is continued to be read by the tRNA, and you'll have all of these amino acids clumping together eventually and held together by a peptide bond. Now, as soon as there's, let's say hypothetically, there's another arginine, let's say there's a proline, and so forth, as they clump together, as soon as there's more than 50 of these strung together, you finally have a protein, and then eventually this reading will be stopped and the protein will be released. And that is the end of translation, and also the end of protein synthesis. So once again, transcription happens in the nucleus when the DNA strand unwinds and unzips, exposing one side of the DNA strand in the nitrogenous bases. Free-floating RNA nucleotides found in the nucleoplasm will then come find their nitrogenous base partner, and as soon as that mRNA string is made, it will then leave the nucleus through the nuclear pore and go out towards the ribosome in the cytoplasm. There it attaches to the ribosome, and then the process of translation starts, where tRNA, the transfer RNA, brings an amino acid that is for that specific codon. As soon as these amino acids find a match, they will start binding with each other through peptide bonds, and as soon as there's more than 50 of them, you will have a protein. So that is protein synthesis. Really not that bad. Just a few things as recap quickly. What is the difference between DNA and RNA? So DNA is a deoxyribose sugar, where RNA is a ribose sugar. The base composition, remember, DNA has thymine, whereas in RNA it is uracil. DNA is a double-stranded molecule, whereas RNA is single-stranded. And then two things that I didn't mention in the previous video is that these bases fall into two groups, purines and pyrimidines. So the purines are the larger group, and this is why they will bond with a different group, the pyrimidines, which are a bit smaller. So adenine will bind with thymine, or it will bond with uracil, and guanine will always bond with cytosine. You don't need to know about purines and pyrimidines, it's just for interest's sake, and this comes down to a rule for those of you that like chemistry, and it's called Chargaff's rule of base pairing, and that is why these nitrogenous bases can only bond with a certain other nitrogenous base. So there is a link in the description tab that will explain that rule just a bit better, but once again, you don't need to know about that rule. You just need to know that adenine will either bond with thymine in DNA, or if it's RNA with uracil, and guanine will always go with cytosine. Another thing they ask in exam papers is to calculate the percentage of a certain nitrogenous base, and that is because with these base pairs there's always a specific ratio. So if you've got adenine bonding with thymine, the ratio will be 1:1, because if there's 30 adenines in a strand, there has to be 30 thymines, because thymine will always bond with adenine. Same thing with guanine. If there's 40 guanines, it means that there has to be 40 cytosines, because the ratio is 1:1. So they can give you a percentage. They'll tell you 34% of the strand is thymine. Then they can ask you what percentage guanine is present. So what you have to do now is you need to do a little bit of maths quickly. So if there's 34% thymine, that means there's going to be 34% adenine, because adenine binds with thymine. So these two together give me a total percentage of, what is that, 68. 68% of the strand is made up of thymine and adenine. Now they want to know what percentage guanine is there. So guanine and cytosine would be the rest, the leftover. So you could say 100 minus 68, because that is what the percentage is of those two, and that should give you, oops, don't know what happened there. So let's try that again. So 100 minus 68 should give you 32%. So this 32% is then both guanine and cytosine. So to get the specific percentage of guanine, you would take 32 and divide it by 2, which will give you 16%, and that 16% is then guanine, and the other 16% would then be cytosine. So there is your answer. So that is also something that they can ask you based on the ratio of these base pairs. So just be prepared. And that is the end of this video. Once again, these videos are just a little bit supplementary if you maybe forgot something that we did in class. Just a quick recap. This should not be the only thing that you use to study this work. It's really just a quick rundown through the work again, just to refresh your memory. But yes, that's the end of the DNA chapter.